The entire system is broken down into three main functional divisions, and this is how we're going to structure our whole discussion. You have the central nervous system, the CNS,
Okay.
then the peripheral nervous system, the PNS,
Right.
and then the one that often gets overlooked but is critically important, the enteric nervous system, the ENS.
The gut brain.
Exactly. Understanding that basic geography is, it's step one. It's the only way to grasp the incredible depth we're about to get into.
Right, so let's start there. Part one, mapping out the system's geography. And the place to begin has to be the headquarters, right? The central nervous system, the CNS.
The command center.
And when we say CNS, what are we physically talking about?
It's actually incredibly simple. It's just two things: the brain and the spinal cord. That's it.
Everything else is outside of that.
Everything else is peripheral. The CNS is the core. It's where all the, the big decisions get made, integration of all incoming information, coordination of all outgoing commands, and of course, all the higher functions we associate with being human.
And even just looking at a cross-section of the brain or spinal cord, you can immediately see it's not uniform. There's a very clear visual difference.
A huge one. And it's probably the first distinction you learn in neuroscience. You have gray matter and you have white matter.
Okay.
And this isn't just a color difference, it's a profound functional difference. Gray matter is where the processing happens. It's packed with the neuron cell bodies, their dendrites, the little input receivers, and all the supporting glial cells.
So that's the computer chip part.
That's the processor, exactly, the integrative cortex.
Which means the white matter must be
the cabling. It's just the transmission lines. It's made up almost entirely of the long myelinated axons that are carrying the signals out of those processing centers, along with the glia that are, insulating those cables.
It's such a logical design.
It is. And you can see this beautiful layered organization in some of the source images. Figure 3.1, for instance, shows a section of the cerebellum.
Right.
And you can see it instantly. There's this incredibly folded, complex outer layer of gray matter. That's the cerebellar cortex. It's where you find those amazing Purkinje cells we'll get to later.
And deep inside them
is the core of white matter, where all the axons from those Purkinje cells are projecting their signals out to the rest of the brain. The design principle is clear. Process on the outside, communicate on the inside.
So that fundamental organization, processing versus communication is key. Okay, so once the signal is processed, it needs to leave the brain or the spinal cord. It steps outside that CNS core.
And now you're in the peripheral nervous system, the PNS.
Which is everything else.
Literally everything else.
Right.
All the nerves that branch out from the brain and spinal cord. If the CNS is the command center, the PNS is the network of conduits, the input and output lines connecting that center to, well, the rest of the body.
And it works both ways. It's bringing information in and sending commands out.
Exactly. Functionally, we talk about sensory input, which is called afferent signaling. Those signals are traveling inward, afferent for arrive.
Okay, arriving at the CNS.
Right. And then you have the motor output, which is efferent signaling. Those signals are traveling outward, efferent for exit.
And that's controlling everything. From me deciding to pick up this pen.
That's the somatic system, conscious muscle movement.
To my heart beating without me thinking about it.
And that's the autonomic system, the involuntary regulation of your organs.
And the architecture, the actual physical structure, it reflects this divide between CNS and PNS. I found this fascinating linguistic detail in the sources about how we name clusters of neuron cell bodies.
Ah, yes. Nuclei versus ganglia.
Exactly. So if you have a bunch of these cell bodies, these little processing hubs, clustered together inside the CNS.
It's called a nucleus, or nuclei for plural.
But the moment that same kind of cluster is found outside the CNS, in the PNS.
It's called a ganglion, or ganglia. It's the same basic idea, a hub of cell bodies doing their metabolic work, but the name changes based entirely on its geographical location.
It's like calling a hill a mountain just because it's in a different country.
A perfect analogy. And a great example is the sensory neurons that bring information from your skin into your spinal cord. Their cell bodies are grouped together just outside the cord in what are called the dorsal root ganglia. They're firmly in PNS territory.
And you mentioned the motor side includes the autonomic nervous system, the ANS. This is the tireless manager working in the background.
It really is. It's the system that keeps you alive without you ever having to think about it. It's all about maintaining internal balance, or what we call homeostasis.
What's it controlling?
Pretty much all the involuntary stuff. Your cardiac muscle, the smooth muscle that lines your blood vessels and your gut and all of your glands. It's the system that, makes your heart race before you've even consciously processed a threat.
And this is where we get that classic division, right? The gas
Yeah.
pedal and the brake.
Exactly. You have the sympathetic division, which is all about mobilizing resources for fight or flight.
Increasing heart rate, diverting blood to muscles.
All of that. And then you have the parasympathetic division, which is the rest and digest system. It conserves energy, manages routine operations like digestion. And these two are in this constant, delicate tug of war to maintain that perfect balance.
Okay, so we've got the CNS as the core, the PNS as the network, but you said there was a third division.
Yes. And we absolutely have to give it its due. This is 1.2 in the outline, the enteric nervous system, or ENS.
The famous gut brain.
The gut brain. And it sounds like a dramatic nickname, but it is physically real. It's this incredibly complex network of neurons that is located entirely within the wall of your gastrointestinal tract.
From top to bottom.
From the esophagus all the way down to the colon. And its significance, the reason it gets called the third division, is its remarkable degree of autonomy.
So it can think for itself, so to speak.
In a way, yes. It controls the entire local environment of the gut. It manages secretions from the gut wall. It regulates local blood flow to aid in absorption. And crucially, it directs motility, the complex, coordinated muscular contractions that move food along.
And it can do this on its own.
That's the key point from the sources. While the main autonomic system, the sympathetic and parasympathetic nerves, certainly influences the ENS can function independently. It can handle these complex local reflexes and regulations, even if its connection to the CNS were completely severed.
Wow. So it's like a franchise manager who can run the whole store without ever calling corporate.
That's a great way to put it. It is a complete, self-contained nervous system dedicated to one very complex job. It has its own sensory neurons, its own motor neurons, its own inner neurons connecting them, and even its own population of specialized glial cells. It's just a masterpiece of local control.
Incredible. So that's the grand map laid out.
Yeah.
Now we have to look at the actual citizens of this map, right? The cast of characters.
Exactly. 1.3, the primary cell types. And it really boils down to two major categories, the neurons and the neuroglia.
And for the longest time, all the attention, all the focus was on the neuron.
Oh, completely. The neuron was the hero. And for decades, there was this prevailing myth, you'd see it in every textbook, that glial cells outnumbered neurons by about 10 to 1.
And that their job was basically just to hold everything in place.
Literally. The name glia comes from the Greek for glue. They were seen as just passive packing material, the insulation, the stuff that filled the gaps.
But that's all changed.
Dramatically. First, newer, more accurate cell counting methods have shown that the ratio is nowhere near a simple 10.1 across the board. It varies wildly depending on the brain region.
So in some places there are more glia, in others fewer.
Precisely. The sources mention older studies, like one from 1968 by Glaga and Johnson, that found ratios as high as 17 to 1 in places like the thalamus. But then you look at a place like the cerebellum, which is just jam-packed with tiny neurons, and the ratio is much closer, maybe only 3 to 1.
But the ratio itself isn't even the main point anymore, is it?
Not at all. The real takeaway, the huge conceptual shift, is that glial cells are emphatically not just glue. The source material makes it crystal clear. They are functionally essential active participants in everything the nervous system does.
So what are they doing?
What aren't they doing? They're managing nutrient support. They're regulating the chemical microenvironment with incredible precision. They're participating in chemical signaling. And they're basically the immune system of the brain.
There's the stage crew.
They are the ultimate stage crew, maintenance team, and security detail all rolled into one. They allow the rock stars, the neurons, to do their job. And we're going to spend a huge chunk of time in Part 4 detailing their specific roles, because without them, the whole machine just grinds to a halt.
Okay, let's turn our focus then to the star player, the neuron. Let's really get into its anatomy and how it works. Starting with 2.1, the neuron's core structure. You have to begin with the soma.
The cell body. This is the metabolic engine of the whole operation.
The power station.
It really is. It's this high-energy headquarters. The soma has to generate and maintain the entire cell, which can be an immense volume, especially when you consider that some axons can be, a meter long or more.
And you can tell just by looking at it under a microscope that it's incredibly active.
Oh, absolutely. The structural evidence is everywhere. You see a huge centrally located nucleus, often with a very prominent nucleolus, which points to massive ribosome production. You see a highly developed Golgi complex for packaging all the products.
But the real giveaway, the classic identifier for a neuron's soma, is something called Nissl granules, right?
Exactly. Nissl granules are essentially the protein factories of the neuron, cranked up to 11. They're these dense stacks of rough endoplasmic reticulum and free ribosomes, and they're constantly churning out the massive amounts of protein the cell needs to maintain its structure and function.
So the presence of these granules is a direct visual indicator of intense synthetic activity.
It is. Think of the neuron as a city that never sleeps. It's always firing, always repairing itself, always needing new parts and fuel. That synthesis is happening right there in the soma, driven by the Nissl bodies.
And it's interesting that the plasma membrane of the soma itself is generally unmyelinated. It's not wrapped in that fatty insulation.
Right, and that hints at its primary role. It's not built for high-speed, long-distance signal propagation like the axon is. Its job is to receive and integrate incoming signals and to run the cell's entire metabolic and synthetic machinery.
Okay, so from that central hub, we have these structures reaching out to receive information, the dendrites.
The primary receptive surfaces. And the first thing you notice about dendrites is how branched they are. It's a phenomenon we call dendritic arborization because they look like the branches of a tree.
And the logic here is pretty straightforward.
Completely. The entire architecture is designed to maximize the surface area available to receive synaptic inputs from other neurons. A single neuron might receive inputs from thousands, or in some cases, hundreds of thousands of other cells.
And it needs a place to put all those connections.
It needs real estate.
Right.
And the dendritic tree provides that. And what's crucial to understand is that while the basic branching pattern is set during development, that tree is incredibly dynamic. It's plastic. It changes throughout your life.
The complexity of that branching is directly tied to the neuron's job, isn't it?
Directly. The more information a neuron needs to integrate and process, the more complex and extensive its dendritic arbor will be.
And this is where we need to zoom in even further to these tiny little projections on the dendrites themselves, the dendritic spines.
Yes, this is a critical detail. These are tiny, often mushroom-shaped protrusions that stick out all over the dendrites. And these little spines are the primary anatomical location for receiving excitatory synaptic inputs.
So the on signals are mostly landing on these spines.
Overwhelmingly. Especially the ones that use the neurotransmitter glutamate. And the insight here is just profound.
What do you mean?
I mean, cognition, learning, memory. It's not just about which neurons are firing. It's housed in the constant physical, morphological changes of these tiny, tiny structures.
So the spines themselves are changing shape.
Constantly. They are highly plastic, their shape, their size, their density, how many there are. It's all in flux, changing in response to the demands placed on the circuit. This is the physical basis of learning.
So when you learn something new, you're literally growing or changing the shape of these spines.
You are. The sources give a great example from the olfactory system. The density of dendritic spines changes based on your exposure to new smells. They aren't fixed receiving docks. They are these transient structures that are constantly being built, remodeled, and pruned away.
So the brain is literally rewiring itself on a moment-to-moment basis.
At the level of individual spines.
Yeah.
Exactly. It's allowing the neural circuit to physically change its configuration based on experience.
Okay, so if the dendrites and their spines are the receivers, we need a dedicated output line, and that's the axon.
The specialized highway for signal propagation. And every axon has a distinct starting point, the axon hillock. You can think of the axon hillock as the final checkpoint. It's this cone-shaped region of the soma where the axon begins, and it's anatomically distinct. Specifically, it completely lacks Nissl granules and Golgi complexes.
Why is that?
Because its job isn't synthesis. Its job is signaling. All the protein manufacturing is done back in the soma. The hillock is where the decision to fire is made. The electrical signal, the depolarization, then hits the initial segment right after the hillock.
And that's the trigger zone.
That's the trigger zone. It typically has the lowest threshold for firing, so this is where the action potential is officially born.
And from there, the axon can just go for huge distances.
For incredible distances. An axon can run for meters in a large animal. Along the way, it can give off side branches, which are called axon collaterals, to communicate with other cells. And it always ends in this complex branching structure called the terminal arborization.
The terminal branches.
Right. And this is where it will make its synaptic connections to the next cells in the chain, other neurons, muscle fibers, or glands.
Now, given these immense distances, how on earth does the cell maintain this highway? The end of the axon is a world away from the factory in the soma.
That is the central logistical problem for the neuron. And the solution is 2.3. Axoplasmic transport. It's this critical internal highway system.
So it's not just an electrical cable, it's a physical supply line.
It has to be. The soma creates all the structural parts, the fuel, the neurotransmitters, and they have to be physically shipped down to the distant terminals. This movement happens along microtubule tracks, like little rail lines, and it's powered by specialized molecular motors.
And it have to go both ways, right?
Absolutely, it must be bidirectional. The movement away from the soma toward the terminal is called anterograde transport.
Okay.
And this is driven by a family of motor proteins called kinesins. You can picture kinesin as this tiny little delivery truck that literally walks its cargo down the microtubule track.
And is all cargo shipped at the same speed?
No, and that's a key distinction. We have fast anterograde transport, which is just astonishingly quick. It moves at up to 400 millimeters per day.
That's incredible. What's in the fast lane?
All the time-sensitive critical components, the things the terminal needs to function right now. So we're talking about synaptic vesicles filled with neurotransmitters, mitochondria to provide energy, and other essential organelles.
And the slow lane.
That's slow anterograde transport, and it's much, much slower, moving at somewhere between 0.2 and 8 millimeters per day.
That's a huge difference. What's it carrying?
It's carrying the heavy freight, the foundational structural components, the massive volume of cytoskeletal elements like neurofilaments and the microtubules themselves that are needed to maintain the physical shape and volume of the huge axon. You can't rush the delivery of the actual railroad tracks.
Okay, so that's the delivery route. What about the return trip?
That's retrograde transport. It's moving material back toward the soma, and it's driven by a different motor protein called dynin. It moves at a pretty brisk pace, about 200 millimeters per day.
And what's it bringing back? Garbage.
That's a big part of it, yes.
Huh.
It's the essential recycling route. It carries old, degraded organelles back to the soma to be broken down and reused. But critically, it's also a communication line. It carries signaling molecules, like trophic factors, from the terminal back to the cell body. These signals provide the soma with crucial feedback about the health and activity state of its distant connection. It's how the soma knows if its terminal is successfully connected and thriving.
And this return pathway, it has a dark side.
A very dark side. As the sources point out, this retrograde transport highway is ruthlessly exploited by certain pathogens.
For example.
The tetanus toxin. It gets into a peripheral wound, gets taken up by a neuron terminal, and then it hijacks the dynin motors to travel all the way up the axon into the spinal cord, where it can cause its devastating, lethal effects.
And viruses, too.
Absolutely. Viruses like herpes zoster, the virus that causes shingles, and rabies, they use this exact same highway system to travel from the periphery into the central nervous system or into the ganglia where they can hide out.
It's just a pathway, and it can be used for good or for ill.
Exactly. And speaking of these molecular motors, the sources make a brief but interesting connection to something called intraflagellar transport, or IFT.
IFT. I associate that with flagella, like on a sperm cell. What's it doing in a neuron?
Well, it turns out that many neurons have what's called a primary cilium. It's not for movement. It's a non-modal little antenna that sticks out from the cell surface.
An antenna for what?
For sensing the local environment. IFT is the transport system that operates inside the cilium, moving proteins up and down. And it's crucial because it connects the neuron to external signaling pathways, like the sonic hedgehog pathway.
And why does that matter?
Because those pathways are fundamental for regulating how neurons develop, how they differentiate, and whether they survive. So the same family of motor proteins that's running the massive freight lines down the axon also has this subtle parallel function in sensing the world right outside the cell body. It's just another incredible layer of complexity.
So we've seen the basic structure, but not all neurons look the same. Let's move to 2.2, this idea of neuronal diversity. Form dictates function.
Absolutely. And the most basic way we classify neurons is by the number of processes or neurites that extend from the soma. It tells you a lot about the cell's job.
Okay, so what's the simplest form?
That would be the unipolar neuron, or more accurately, the pseudo-unipolar neuron. These are almost exclusively sensory neurons.
Like the ones in the dorsal root ganglia we mentioned.
Exactly like them. They start out in development with one process that then splits in two. One branch goes out to the periphery, to your skin or muscles, to act as the sensory receptor. The other branch goes into the CNS to deliver that signal. It's a single, continuous line for transmitting sensory information.
Okay, so one process that splits. What's next?
Bipolar neurons. These are much rarer, and you only find them in very specialized sensory pathways. Think the retina of the eye, the olfactory epithelium in your nose, and parts of the inner ear.
And bipolar means two processes.
Exactly. One single dendrite on one side of the soma and one single axon on the other. It creates this very direct, straight-line flow of information. They're specialists.
Which leaves the most common type.
The vast majority of neurons, especially in the CNS, are multipolar neurons. These are the great integrators. They're defined by having one axon and two or more dendrites.
So they can receive input from many different sources.
Many, many sources. This category includes everything from the giant motor neurons in your spinal cord that control your muscles to the incredibly complex cells of the cerebral cortex.
And the source material highlights a couple of key examples of this multipolar specialization.
It does. There are the pyramidal cells of the cortex, which are named for their triangular cell body and are absolutely vital for learning and higher cognitive functions.
But the one that's just visually breathtaking is the Purkinje cell.
The Purkinje cell is an anatomical masterpiece. You find it in the cerebellar cortex, and its defining feature is this massive, flat, almost two-dimensional dendritic tree. It's like a giant fan.
And it's just covered in synaptic inputs.
Thousands upon thousands of them. It makes the Purkinje cell one of the most powerful integrators in the entire nervous system. And you see similar complexity in other cells, like the fluorescent dye image of a retinal ganglion cell in figure 3.3. The point is, these aren't just simple on-off switches. They are incredibly intricate, highly specified biological circuits.
All right, so all of this amazing structure, the soma, the dendrites, the axon, the transport systems, it's all in service of one single critical fleeting moment.
The synapse.
The synapse. Let's dive into part 3 and really spend some time on this, starting with 3.1, the basic structure of the synaptic junction.
Okay, so the synapse is simply this specialized site where one cell passes its signal to the next. And in the mammalian nervous system, this happens overwhelmingly through a chemical synapse.
And what defines a chemical synapse?
The key feature is a physical gap. There's no direct connection. The two cells are separated by a tiny space called the synaptic cleft, which is typically about 20 to 30 nanometers wide.
Not very big, but it's a real gap.
It's a real gap. And it means the electrical signal can't just jump across. It has to be converted into a chemical signal to cross the space and then converted back into an electrical one on the other side.
Now, we're focusing on chemical synapses, but the sources do mention that there's another kind.
There is. We have to acknowledge the electrical synapse.
How does that work?
Electrical synapses use structures called gap junctions. These are basically direct protein channels that connect the cytoplasm of the two cells. The cleft is much smaller, only about 2 nanometers, and it allows electrical current to flow directly from one cell to the next.
So it's much faster.
It's virtually instantaneous. There's no delay for chemical conversion. They're really important for synchronizing the activity of large groups of neurons, for instance, in the parts of the brainstem that control rhythmic things like breathing. But they are much rarer than chemical synapses in the mature CNS.
Okay, so back to the chemical synapse. The sources provide a really useful classification system that helps you predict what a synapse does just by looking at its structure. Type I versus type 2.
Yes, this is a really powerful anatomical insight. Type I synapses are, as a rule, excitatory. They turn the next cell on.
And what do they look like?
Structurally, they're described as asymmetrical. And what that means is that the postsynaptic side, the receiving side, has this thick, dense-looking layer of proteins. It's called the postsynaptic density, or PSD, and it shows up as a dark band under the electron microscope.
Like it's heavily fortified for receiving a signal.
That's a great way to think of it. It's packed with receptors and scaffolding proteins. The vesicles on the presynaptic side of a type I synapse are also typically small, clear, and spherical, and they're usually filled with the excitatory transmitter glutamate.
Okay, so asymmetrical and excitatory. That must mean type 2 is the opposite.
Exactly. Type 2 synapses are typically inhibitory. They turn the next cell off, and structurally, they are symmetrical.
Meaning the postsynaptic density is much thinner.
Much thinner and less pronounced. And the vesicles on the presynaptic side are often described as being flattened or pleomorphic, meaning they have irregular shapes. These are often filled with inhibitory transmitters like GABA or glycine.
And the location of these synapses on the neuron is also very deliberate, isn't it?
Oh, incredibly deliberate. The most common arrangement is the axidendritic synapse, where an axon connects to a dendrite. And very often, these are type I excitatory synapses landing on those plastic dendritic spines we talked about.
But inhibition is different.
Inhibition often requires more direct control over whether the cell fires or not. So you often find axosomatic synapses, where an axon connects directly to the cell body, the soma. These are very often type 2 inhibitory synapses,
Why?
right? Because if you deliver a powerful off signal right at the cell's headquarters, right next to the axon hillock where the decision to fire is being made, you can effectively veto all the incoming on signals from the dendrites. It's a position of immense power.
And there's one more highly specialized type, the axoaxonic synapse.
Right. An axon terminal connecting to another axon terminal. This is a bit more complex. It's the basis for a mechanism called presynaptic inhibition. It allows one neuron to selectively control how much neurotransmitter another neuron releases.
So it's not turning the whole cell off, just turning down the volume on one specific input.
Precisely. It's an incredibly fine-tuned level of control.
Okay, let's walk through the actual event. 3.2, the synaptic activation mechanism. How does this signal relay happen so fast?
It all begins with the arrival of the electrical signal, the action potential, at the presynaptic terminal.
Right.
The wave of depolarization from the action potential hits the terminal, and this change in voltage immediately causes specialized voltage-gated calcium channels in the terminal's membrane to fly open.
So calcium is the key.
Calcium is the trigger, the immediate essential trigger. There's a much higher concentration of calcium outside the cell than inside, so when these channels open, calcium ions flood into the terminal.
And what does that calcium influx do?
It gives the final command. It triggers this complex protein machinery, the snare proteins, that causes the synaptic vesicles, which are already docked and waiting at the membrane, to fuse with it.
And then they release their contents.
They release their neurotransmitter content into the synaptic cleft in these discrete little packets called quanta. The entire process, from calcium entry to transmitter release, happens in fractions of a millisecond. It's an absolute miracle of coordination.
But if a neuron is firing rapidly, it's going to run out of vesicles pretty quickly.
It would if it didn't have an incredibly efficient recycling system. And the sources really emphasize the importance of this. Vesicle recycling is critical.
So how does it get the empty vesicles back?
Once the transmitter is released, the membrane of the vesicle, which is now part of the presynaptic membrane, is rapidly retrieved via a process called endocytosis. This is often mediated by a protein called clathrin, which helps pull the membrane back in.
And then it gets refilled.
It gets reformed into a new, empty vesicle right there in the terminal, gets refilled with neurotransmitter, and is ready to be used again. This rapid renewal means the neuron can keep up a constant supply of ready-to-go packets, allowing it to sustain high-frequency firing without running out of ammo.
So the transmitter is now in the cleft.
Yeah.
What happens next depends entirely on the receptor on the other side.
Entirely. And we have two major classes of receptors, and they dictate the speed and nature of the response. The fast track is the ionotropic receptor.
Ionotropic, what does that mean?
It means the receptor is itself an ion channel. It's a ligand-gated channel. When the neurotransmitter binds to it, the channel part of the protein just pops open directly.
And ions flow through.
And ions like sodium or chloride or potassium flow through, causing an immediate rapid change in the membrane potential of the postsynaptic cell. The effect is measured in milliseconds. It's for fast, short-term communication. It's like a simple light switch.
But it's not the only way.
No. The other class is the metabotropic receptor, and this is the long game.
How does it work?
These are G protein-coupled receptors. When the neurotransmitter binds, it doesn't open a channel directly. Instead, it activates a G protein inside the cell, which then triggers a whole cascade of intracellular second messengers.
So it's an indirect effect.
A very indirect effect. The response is much slower. It might take seconds or even minutes to develop, but the effects are often much more profound and long-lasting. It can lead to massive changes inside the cell, altering enzyme activity, or even turning genes on or off. This is neuromodulation.
Okay, this brings us to the actual chemical alphabet itself. 3.3, the diversity of neurotransmitters. This is where the complexity really explodes.
It really is. The nervous system uses this huge variety of chemicals to send messages with different speeds, different durations, and different effects.
Where should we start?
Let's start with a classic, acetylcholine, or HE. It's historically important, the first neurotransmitter to be discovered, and it's functionally vital.
This is the one that controls our muscles, right?
It's the primary transmitter at the neuromuscular junction, yes, but it's also used extensively throughout the autonomic nervous system and in the brain for things like attention and memory.
And it uses different kinds of receptors.
It does. It has two main subtypes. The nicotinic receptors are ionotropic. They're the fast-acting ones found on skeletal muscles. When ATEC binds, they pop open a channel that lets sodium in, causing the muscle to contract.
In the O-type.
Muscarinic receptors. These are metabotropic, the slower G protein-coupled ones. You find them on smooth muscle, cardiac muscle, and glands, where they mediate slower, more modulatory effects.
And with something as critical as muscle contraction, you need to be able to turn the signal off just as quickly as you turn it on.
Instantly. And the action of ACHIC is terminated by a super-efficient enzyme in the synaptic cleft called acetylcholinesterase, or ACE. It just shreds the ACHIC molecule, deactivating it almost immediately.
And this is why nerve agents are so deadly.
Exactly. Nerve agents like sarin gas and many insecticides work by irreversibly blocking ACE. So ACE builds up in the synapse, leading to constant, uncontrolled muscle contraction. The result is spasms, paralysis, and ultimately death.
And on the flip side, you have diseases like myasthenia gravis.
Right, where the body's own immune system produces antibodies that block the nicotinic receptors at the neuromuscular junction, leading to profound muscle weakness because the signal can't get through properly.
Okay, so after acetylcholine, let's move to the biogenic amines.
Right. These are often called neuromodulators. They don't typically mediate the fast point-to-point signaling. Instead, they tend to regulate overall brain states like mood, attention, and arousal.
So they're setting the background tone.
That's a perfect way to put it. And the big ones here are norepinephrine, also known as noradrenaline, and adrenaline.
This is the fight-or-flight stuff.
Peripherally, yes.
Yeah.
Norepinephrine, or NE, is released by the sympathetic postganglionic neurons to mobilize the body. But in the CNS, it's produced by a tiny little nucleus in the brainstem called the locus coeruleus.
And that one nucleus has a huge impact.
A massive impact. It sends its axons out to virtually the entire brain and spinal cord, and it's critical for regulating arousal, vigilance, and mood.
Okay, then we have dopamine, or DA.
Dopamine is a huge player. It governs two really critical systems. First, a pathway originating in the substantia nigra is essential for initiating movement.
Which is why its loss is so devastating in Parkinson's disease.
Precisely. The death of those dopamine-producing neurons in the substantia nigra is the direct cause of the motor symptoms of Parkinson's. The second major system is the mesolimbic pathway, which is all about reward, motivation, and behavior.
The feel-good chemical.
It's often called that, yes. It's central to how we learn to repeat behaviors that are rewarding.
And the third major amine is serotonin, or 5-HT.
Right. Serotonin is produced primarily by a collection of nuclei called the Raphi nuclei, and like the locus coeruleus, they project incredibly widely throughout the brain.
And what does serotonin influence?
Almost everything. Sleep-wake cycles, appetite, mood stability, aggression. Its influence is so vast, which is why it's the target for so many medications used to treat depression and anxiety, the SSRIs.
Okay, so those are the modulators. What about the real workhorses of moment-to-moment communication?
For that, we have to turn to the amino acids. These mediate the overwhelming majority of all the fast, excitatory, and inhibitory signaling in the entire central nervous system.
And the king of the on signals is?
Glutamate. Without a doubt. Glutamate is the dominant excitatory neurotransmitter in the CNS.
And like acetylcholine, its function is really defined by its different receptor subtypes.
It is. You have the AMPA and kainate receptors. These are both straightforward ionotropic receptors. Glutamate binds, they open, sodium rushes in, and the cell is excited. Simple, fast activation.
But then there's a special one, the NMDA receptor.
The NMDA receptor is the star of the show. It's unique because it's both ligand-gated, it needs glutamate to bind to it, and it's voltage-dependent.
What does that mean, voltage-dependent?
It means that at the normal resting membrane potential, the channel of the NMDA receptor is physically blocked by a magnesium ion. It's like a cork in a bottle.
So even if glutamate is there, nothing happens.
Nothing happens. The cork only gets removed, the magnesium ion only pops out if the membrane is already partially depolarized, usually by the action of nearby AMPA receptors.
So you need two things to happen at once.
You need glutamate to be present, ANA the cell to be active. And when that happens, the NMDA receptor opens and allows not just sodium, but critically, calcium to flow into the cell.
And we know that calcium is a powerful internal signal.
It's transformative. That influx of calcium acts as a potent second messenger that triggers long-lasting changes inside the postsynaptic cell. It strengthens that specific synapse in a process we call long-term potentiation, or LTP.
Which is the cellular basis for learning and memory.
It is. This is how a memory trace is physically encoded in the brain. But this power comes with a very serious risk. The same mechanism that enables learning can also kill the neuron. If you have excessive uncontrolled release of the glutamate, like what happens during a stroke or a severe head injury, it leads to massive, prolonged activation of NMDA receptors.
And a flood of calcium into the cell.
A toxic flood of calcium. This is a phenomenon called excitotoxicity. The neuron is literally excited to death. It's a double-edged sword that's built right into the core of the system.
So that's the main on switch. What's
Yep.
the main off switch?
There are two, really. The main inhibitory transmitters are GABA, which stands for gamma-aminobutyric acid, and glycine.
And how do they work?
They typically bind to ionotropic receptors that are channels for chloride ions. When they open, negatively charged chloride ions flow into the cell, which makes the inside of the cell even more negative.
It hyperpolarizes it.
Exactly, which moves it further away from the threshold for firing, making it much less likely to generate an action potential. GABA is the main player throughout the brain. You find it everywhere, especially in those little inhibitory interneurons that fine-tune circuit activity.
And glycine.
Glycine does pretty much the same job, but it's primarily found in the spinal cord and the brainstem. And clinically, this is the system that many sedative drugs target. Things like benzodiazepines or alcohol work by enhancing the effect of GABA, leading to a general reduction in overall neural excitability.
Okay, so we've covered the small, fast-acting molecules. But there's another class, right? The big ones.
Yes, the neuropeptides, these are a whole different ballgame. They're much larger molecules packaged in large, dense core vesicles.
And they're not the primary signal.
No, they act mostly as neuromodulators. They're often co-released along with one of the small molecule transmitters, and their job is to produce slower, longer-lasting, more diffuse effects.
And they're made differently, too.
They are. Because they're proteins, they have to be synthesized back in the soma and then shipped all the way down the axon via that fast transport system. This means their release and replenishment is a much slower process.
What are some key examples?
Well, there's substance P, which is intensely involved in transmitting pain signals, especially that slow-burning kind of pain. There's VIP, or vasoactive intestinal peptide, which is important in the gut and for regulating blood vessels.
And the body's own painkillers.
The opioids, like the enkephalins and endorphins, they function as the body's natural pain modulators. They work by inhibiting the release of other transmitters, like substance P, effectively dampening pain signals before they even reach the brain.
Okay, and just to be thorough, the sources mention a couple of other oddballs.
A few, yes. We have to mention purinergic signaling, which uses ATP and adenosine. ATP, the energy currency of the cell, can also act as a co-transmitter. And its breakdown product, adenosine, is an important inhibitory signal that builds up while you're awake and contributes to the feeling of sleepiness.
So that's what caffeine blocks.
That is exactly what caffeine blocks. It's an adenosine receptor antagonist.
And the last one is really strange.
Nitric oxide, or NO, it just breaks all the rules.
Yeah.
First, it's a gas. Second, it's not stored in vesicles. It's synthesized on demand in the postsynaptic neuron. And because it's a gas, it can just diffuse freely across cell membranes.
It can travel backwards.
It can. It often acts as a retrograde messenger, diffusing back from the postsynaptic cell to the presynaptic terminal to influence its activity. It's involved in long-term potentiation and in regulating local blood flow to match neural activity. It's a truly unconventional signal.
Wow. Okay, so we spent a lot of time on the neuron, but we said at the start that none of this would be possible without its support crew. So let's dive deep into part 4, the glial support system and protective barriers. And we have to start with the most abundant ones, the astrocytes.
The CNS housekeepers.
Yeah.
And they really are. They're the most numerous type of glial cell in the CNS, named for their star-like shape, and they're identified by a protein they express called GF8.
And their job description is long.
It's incredibly long and diverse. They are the ultimate mediators. Their little end-feet processes connect to neurons, to blood vessels, and to the protective outer layers of the brain. They're in the middle of everything.
Okay, so give me the highlight reel. What are their most critical tasks?
Okay, number 1, structural support is part of it. But much more importantly, they are the absolute masters of regulating the chemical microenvironment. A key example is potassium ion buffering.
What does that mean?
When neurons fire action potentials over and over, they release potassium ions, K plus A, into the tiny space outside the cell. If that potassium builds up, it changes the electrical gradient and can cause all the surrounding neurons to start firing uncontrollably.
So the astrocytes clean it up.
They soak it up. They rapidly absorb this excess potassium and then shuttle it through their own network of interconnected cells, dispersing it safely and maintaining the stability that the system needs for precise signaling.
That's incredible. What's next?
They are the master recyclers of neurotransmitters, especially glutamate. The glutamate clearance mechanism is absolutely essential for life.
Why?
Because of excitotoxicity. If glutamate just hung around in the synapse, it would keep stimulating the postsynaptic neuron to death. Astrocytes have these powerful transporters that suck the glutamate out of the synaptic cleft almost instantly.
And then what do they do with it?
They convert it into an inactive form called glutamine. Then they shuttle that glutamine back to the neuron, which can then safely convert it back into glutamate to be used again.
So successful communication isn't just about the signal being sent. It's about the astrocyte immediately cleaning up the mess.
Immediately and completely. The cleanup is as important as the signal itself. And on top of that, they provide vital metabolic support.
How so? They feed the neurons.
In a way, yes. Neurons are incredibly energy hungry. Astrocytes have their end feet right on the blood capillaries. They absorb glucose from the blood, convert it into lactate, and then shuttle that lactate directly to the neurons as a ready-to-use fuel source, especially during periods of high activity.
They're basically running a catering service.
A high-end, personalized catering service.
That.
And all of this functional intimacy, the potassium buffering, the glutamate recycling, the metabolic support, it all leads to this really important modern concept of the tripartite synapse.
The three-part synapse.
Exactly. We used to think of a synapse as just being the presynaptic terminal and the postsynaptic dendrite, a two-part conversation.
But the astrocyte is the third party.
It is. We now know that an astrocyte process intimately wraps around that entire junction. It's actively listening in, it's monitoring the levels of neurotransmitters, and it's releasing its own signaling molecules, called gliotransmitters, to actively regulate that synapse's strength and plasticity.
So the astrocyte is an active participant in the conversation. It's not just a passive observer.
Not at all, and this makes them hugely important in disease. When there's an injury to the brain, like a stroke or trauma, astrocytes go into overdrive. They proliferate and get bigger in a process called reactive astrogliosis.
They form a scar.
They form a glial scar. And initially, this is protective. It walls off the damaged area. But long-term, that dense scar can actually prevent axons from regenerating and making new connections.
And you said their end feet are on the blood vessels.
Completely sheathing them, which makes them a crucial physical component of the blood-brain barrier, which we'll get to in a moment. They're just fundamentally important to everything the CNS does.
Okay, so if astrocytes are the housekeepers, let's talk about the electricians. 4.2, the myelin-forming glia. The insulators.
Right, the cells that make high-speed communication possible. And this job is handled by two different types of cells, depending on whether you're in the CNS or the PNS.
Let's start with the CNS.
In the central nervous system, the job falls to the oligodendrocytes. And the architectural genius of the oligodendrocyte is its efficiency.
What do you mean?
A single oligodendrocyte can myelinate multiple axons. It sends out these long, flat processes, like arms, and each arm wraps itself around a segment of a nearby axon, forming the myelin sheath. One cell can insulate up to 50 different axonal segments.
It's a one-to-many relationship.
A one-to-many relationship. It's incredibly efficient.
But it's different in the PNS.
It is. In the peripheral nervous system, the job is done by Schwann cells, and their approach is much more dedicated and perhaps simpler. A single Schwann cell is responsible for myelinating only one segment of one axon.
A one-to-one relationship.
Exactly. The Schwann cell wraps its entire body around that one segment. They also have another job, though. They support the small, unmyelinated axons by bundling a group of them into little grooves on their surface in a structure called a musaxon.
In the myelin itself, what is it made of?
It is mostly fat. The chemistry of myelin is what gives it its insulating power. It's about 70 to 80% lipid, which provides that necessary electrical resistance.
But the proteins are different, CNS versus PNS.
Yes, and this is very important clinically. The specific proteins that help compact the myelin sheath are different. In the CNS, you have proteins like PLP and MAG. In the PNS, the major components are proteins called P0 and P2.
And why does that difference matter?
Because it dictates which diseases attack which system. Multiple sclerosis, MS, is a disease of the CNS. The immune system specifically attacks the oligodendrocytes and their myelin. It doesn't touch the PNS.
Where something like Guillain-Barré syndrome
Is primarily a disease of the PNS,
Yeah.
where the immune system often targets the Schwann cells and the PNS myelin. That chemical difference leads to completely different clinical pictures.
And this whole system of insulation, of high-speed transmission, it only works because of the gaps.
It only works because of the gaps, which brings us to 4.3, the nodes of Ranvier and the structures around them. The nodes are the entire key to what we call saltatory conduction.
So what exactly is a node of Ranvier?
It's the tiny bare patch of axon membrane that's exposed between two segments of myelin sheath. And these nodes are like specialized electrical refueling stations.
How so?
They are packed, jam-packed, with an incredibly high density of voltage-gated sodium channels.
So when the electrical signal, the wave of depolarization, is traveling down the axon, it can't leak out through the fatty myelin.
Right. It's forced to jump, essentially, from one node to the next. When the signal arrives at a node, it triggers that massive cluster of sodium channels to open, which instantly and powerfully regenerates the action potential to its full strength.
And then it jumps to the next node.
And then it jumps to the next. This jumping, or saltatory conduction, allows the signal to travel exponentially faster than it could if it had to be regenerated at every single point along the axon, like in an unmyelinated fiber.
But it's not just the node itself that's important. The area right next to it is also highly structured.
Critically so. The regions right next to the node, called the paranodal and juxtaparanodal regions, are where the myelin sheath is physically anchored to the axon.
And what do we find there?
This is where we find clusters of specialized potassium channels, specifically Kv 1.1 and 1.2. And more importantly, this is where we find the adhesion molecules that form the seal.
The seal.
Yes, proteins like Casper and NF155. You can think of them as tiny biological staple guns. They form this tight junction that physically staples the edge of the myelin sheath to the axon's membrane, the axillemma.
And why is that seal so important?
Because it prevents the electrical current from leaking out from under the myelin. It ensures all the current is funneled directly to the next node where it can do its job. If that seal breaks down, the current leaks, the signal slows, and eventually the conduction will fail entirely. It's an incredible example of how a huge function, like moving your hand quickly, depends entirely on the precise organization of a few proteins at the nanometer scale.
Okay, we've covered the housekeepers and the electricians. Now we need to talk about the security guards. 4.4. The microglia.
The resident immune watchdogs of the CNS. And they are functionally very different from the other glia. They don't come from the same neural stem cells. They actually originate from precursors in the yolk sac very early in development and migrate into the brain.
So they're a completely different lineage.
A completely different lineage. They are the brain's dedicated immune system.
And what do they do all day?
In their normal, healthy state, their quiescent state, they are constantly surveying their environment. They adopt what's called a ramified morphology. They have a small cell body with these very fine, constantly moving, branching processes that are touching all the synapses and cells around them.
They're on patrol.
Constantly on patrol, looking for any sign of trouble, an infection, an injury, cellular debris.
And what happens when they find it?
When they detect trouble, usually through inflammatory signals, they undergo a rapid and profound transformation. They pull in their branches, their cell body gets much larger, and they adopt what's called an amoeboid form.
Why do they change shape?
Because their function changes. In this activated amoeboid state, they become migratory. They can move towards the site of injury, and they become intensely phagocytic.
Meaning they eat things.
They eat things, they're the cleanup crew. They engulf and destroy invading pathogens, they clear away the debris from dead or dying neurons, and there's growing evidence that they also play a role in pruning weak or unnecessary synapses during development and learning.
So their role is huge.
Massive. They are absolutely critical for maintaining brain health. But like any immune system, they can sometimes go awry and contribute to autoimmune diseases or the chronic inflammation we see after an injury.
All right, let's move upward a bit, structurally, to 4.5, the system that produces and circulates the fluid that bathes the entire CNS, the ependymma, and the choroid plexus.
Right, the cerebrospinal fluid, or CSF. This is the fluid that fills the brain's ventricles and surrounds the brain and spinal cord, acting as a cushion and a medium for transport.
It starts with the ependymal cells.
The ependymal cells are the epithelial cells that form the lining of the ventricles and the central canal of the spinal cord. In many places, they have cilia on their surface.
And the cilia beat to move the fluid around.
Exactly. The coordinated beating of these cilia helps to circulate the CSF throughout the system. These cells form a barrier, the blood-CSF barrier, but it's sometimes described as a bit leaky or incomplete, allowing for some passive flow of substances.
But the real manufacturing plant for CSF is the choroid plexus.
That's where the vast majority of it is actively produced. The choroid plexus is this specialized tissue found within the ventricles. It's made of these highly vascularized, cauliflower-like folds of pia mater, the innermost protective layer of the brain, and these folds are covered by a layer of specialized cuboidal ependymal cells.
And what makes these cells so special?
They are metabolic powerhouses, just packed with mitochondria and other organelles. And most importantly, they are joined together by very strong, continuous tight junctions.
And that's the key.
That is the key. Those tight junctions form the true, robust blood-CSF barrier at this location. It means that CSF is not just a simple filtrate of blood plasma. It is actively created by these specialized cells, which use powerful transporters to very carefully pump specific ions and molecules out of the blood and into the ventricles, creating this highly controlled fluid environment for the brain.
Which brings us to the ultimate security system. 4.6, blood-brain barrier, the BBB.
Right. This is the barrier that maintains the CNS's incredibly stable and isolated chemical environment, protecting it from all the fluctuations and potential toxins in the general bloodstream.
And what's it made of, physically?
The primary structure of the BBB is formed by the endothelial cells that line the capillaries inside the CNS. Now, everywhere else in your body, the capillaries are a bit leaky. There are gaps between the cells.
But not in the brain.
Not in the brain. In the CNS, these endothelial cells are sealed together by exceptionally strong and continuous tight junctions. These junctions completely block the paracellular route, the pathway between the cells.
And it's more than just the endothelial cells, isn't it?
It's a multi-layered barrier. Those sealed endothelial cells are then wrapped by contractile cells called pericytes, and then the entire structure is completely ensheathed by the specialized end feet of astrocytes.
So it's a 3-layer security fence.
At least. And this combination creates a formidable, highly selective blockade.
How selective is it?
Extremely. The BBB will only allow small, highly fat-soluble lipophilic molecules to diffuse across the membranes. Everything else, including vital things like glucose and amino acids, can only get across if it has a specific, dedicated transporter protein to carry it.
And this is a huge problem for medicine.
A massive clinical challenge. It's great at keeping bad things out, but it's also incredibly effective at keeping therapeutic drugs out, which makes treating brain diseases very, very difficult.
And what happens if the barrier breaks down?
The clinical impact is immediate and severe. If those tight junctions fail due to a head trauma, a severe infection, or a brain tumor, substances that are normally excluded from the brain, like large blood proteins or immune cells, can leak into the brain tissue.
And that causes huge problems.
Huge problems. It can trigger massive inflammation, swelling, edema, and widespread cellular damage. The integrity of the BBB is absolutely non-negotiable for a healthy brain.
Okay, let's shift our focus now from the central command out to the network itself. Part 5. Peripheral nerves and sensory receptors. Let's start with 5.1, the actual structure of a peripheral nerve. It's not just a bundle of axons, it's a highly organized cable.
It's a beautifully organized and protected structure. A peripheral nerve is bundled together by 3 distinct layers of connective tissue.
Okay, let's start from the outside in.
The outermost layer is the epineurium. This is a thick, dense, tough layer of connective tissue that wraps around the entire nerve. It's the protective outer jacket, providing mechanical strength and cushioning.
And inside that?
Inside that, the axons are bundled into groups called fascicles, and each fascicle is wrapped by the next layer, the perineurium. And this layer is functionally critical.
Why is the perineurium so important?
Because the flattened cells that make up the perineurium are joined together by tight junctions.
Ah, so it's another barrier.
It's another barrier. This creates the essential blood-nerve barrier. It functions a lot like the blood-brain barrier, protecting the delicate axons inside the fascicle from toxins and fluctuations in the general circulation.
And then we zoom into the individual fibers.
Right. Inside each fascicle, surrounding each individual nerve fiber, which is the axon and its associated Schwann cell, is the innermost layer, the endoneurium. This is a very delicate, loose, connective tissue that's rich in capillaries, providing the local blood supply. The whole arrangement is just a masterclass in protective engineering.
And within that protected cable, we have different types of wires. 5.2, the classification of nerve fibers.
Yes. And we classify them based on two things that are very tightly correlated: the diameter of the axon and the degree of myelination.
And the simple rule is,
The bigger the diameter and the thicker the myelin sheath, the faster the signal travels. The conduction velocity is directly related to those physical properties.
and this gives us our functional classes. Let's start with the fastest.
The A fibers. These are the Ferraris of the nervous system. They're large in diameter, heavily myelinated, and their conduction velocities are just incredible, ranging from about 12 up to 120 meters per second.
So what kind of information needs to travel that fast?
Critical time-sensitive information. The motor commands going out to your skeletal muscles, those are A alpha fibers, the absolute fastest. Also, the signals for fine touch, proprioception, knowing where your limbs are, and that first sharp initial pain signal, that's A delta.
You touch something hot, and that's the A delta fiber firing.
That's the immediate ouch, pull your hand back signal. Its speed is essential for reflexes.
Okay, so that's the fast lane. What's in the middle lane?
The D fibers, they're intermediate in speed, conducting at about 3 to 14 meters per second. They're still myelinated, but they're smaller in diameter. You find these primarily as the preganglionic fibers in the autonomic nerve system.
So they're carrying the initial command in an involuntary process.
Exactly. And finally, we have the slow lane, the C fibers,
And these are the slowest.
by far. They crawl along at only 0.5 to 2 meters per second. And the reason is that they are small in diameter, and crucially, they are unmyelinated.
And what do they carry?
They carry the signals that don't need instantaneous response. Things like crude temperature sensation, and most importantly, that slow, dull, throbbing, burning pain that comes after the initial sharp injury.
So the difference in speed between A delta and C fibers is why you feel that initial sharp jab, and then a moment later, the dull ache starts to set in.
That is precisely why. You are feeling your nervous system's different fiber types firing in real time. C fibers also make up most of the postganglionic autonomic fibers.
Okay, so those are the wires. Now let's talk about the sensors at the end of the wires. 5.3, the sensory receptor types. These are the transducers.
Exactly. Their job is to convert some form of energy from the outside or inside world mechanical pressure, temperature, light into the electrical language of the neuron, a graded potential.
And the simplest form are the free nerve ending.
Right. They are structurally the simplest and the most widespread. They don't have a fancy capsule. Their endings just terminate freely in the skin or internal organs, but they're multi-talented.
What do they detect?
They act as nociceptors, which detect pain or noxious stimuli. They act as thermoreceptors, detecting temperature. And they also respond to crude touch.
But where the specialization gets really amazing is in the encapsulated endings, the mechanoreceptors.
Yes. These have these elaborate cellular capsules around their nerve endings that are specifically tuned to detect different types of physical stimuli. And a key way to understand them is by looking at how quickly they adapt to a stimulus.
Okay, let's start with a rapidly adapting one.
The Meissner's corpuscle. You find these very close to the skin surface in the dermal papillae. And being rapidly adapting, or RA, means they fire a burst of signals when a stimulus is first applied, but they quickly shut up if the stimulus is held constant.
So they detect change.
They detect change. They're perfect for detecting light touch, flutter, and low-frequency vibration. They're what you use to feel the texture of a fabric or to notice an object slipping from your grasp.
Okay, what's another one? The really striking-looking one?
The Pacinian corpuscle. These are incredible. They're much larger, found deeper in the skin, and structurally they look like a sliced onion with all these concentric layers or lamellae.
And they are very rapidly adapting.
Very rapidly adapting. Their onion-like structure is the key to their function. The layers are filled with fluid, so they act like shock absorbers. They're completely insensitive to steady pressure. They only fire when those layers are suddenly shifted or deformed.
So they're perfect for detecting.
High-frequency vibration and deep pressure. They're what let you feel the vibration of a tuning fork or a power tool.
And then you have the opposite, a slowly adapting receptor.
Right, like the Ruffini endings. These are also found deep in the dermis. And being slowly adapting, or SA, means they continue to fire as long as the stimulus is present.
What do they respond to?
They respond to sustained pressure and skin stretch. They provide continuous feedback about things like joint angle and the shape of an object you're gripping.
So you have detectors for the start of a touch, detectors for vibration, and detectors for sustained pressure. It's a complete toolkit.
A complete toolkit for interpreting the physical world.
Now let's move from the outside world to our internal world. 5.4, the proprioceptors,
Right.
the body's internal awareness system.
Right. This is the system that tells your brain where your limbs are in space without you having to look. It's essential for any kind of coordinated movement, and it relies on two incredibly specialized sensory structures.
Let's start with the muscle spindles.
Okay. Muscle spindles are these amazing sensory organs that are located inside your skeletal muscles, arranged in parallel with the main muscle fibers.
And their job is to detect what?
They detect muscle length and, just as importantly, the rate of change of muscle length. How fast is the muscle being stretched?
And the spindle itself is a complex little organ.
It is. It has its own specialized little muscle fibers inside it called intrafusal fibers. And it has two different kinds of sensory endings that wrap around them.
What are they?
You have the primary ending, also called the annulus spiral ending. It wraps around the very center of the intrafusal fibers, and it's rapidly adapting. It's the part that's exquisitely sensitive to the speed of the stretch.
And the second one?
The secondary ending, or flower spray ending, it's located a bit off to the side, and it's slowly adapting. It's the part that provides continuous information about the steady, static length of the muscle. So you get both dynamic and static feedback from this one sensor.
It's incredible. And what's the second type of proprioceptor?
The Golgi tendon organ, or GTO.
Yeah.
And its location tells you its function. It's located in series with the muscle at the junction where the muscle connects to its tendon.
So it's not measuring length.
It's not measuring length, it's measuring tension.
Yeah.
It detects how hard the muscle is pulling, the force of its contraction, and functionally, it's slowly adapting.
And this is a protective mechanism.
A hugely important one. While the muscle spindle is telling the brain how long the muscle is, the GTO is telling it how hard it's working. If the muscle tension becomes so high that it risks tearing the tendon, the GTO fires a powerful signal that actually triggers a reflex to inhibit the motor neuron.
It forces the muscle to relax.
It forces an immediate relaxation to prevent a catastrophic injury. It's the body's emergency brake.
So let's bring it all together. Part 6, the actual electrical signal and the motor output. We have to quickly recap 6.1, the action potential, the nerve impulse itself.
Right, the fundamental unit of long-distance communication. And it all starts from the resting state.
The cell is just sitting there polarized.
Exactly. The inside of the membrane is negatively charged relative to the outside, typically around negative 70 millivolts. And this resting potential is maintained by two things. The membrane is much more leaky to positive potassium ions leaving the cell than to positive sodium ions entering, and the constant work of the sodium-potassium pump, which is always pumping 3 sodiums out for every 2 potassiums it punks in.
So it's an active, energy-consuming state.
A very active state. And to fire an action potential, the incoming signals, those graded EPSPs from the dendrites, have to add up. They have to summate at the trigger zone, the axon hillock.
And if they add up enough to reach the threshold?
Then it's go time. Reaching the threshold potential triggers the massive, rapid opening of all the voltage-gated sodium channels in that area. Sodium floods into the cell, and the membrane potential skyrockets. That's the rapid depolarization, the rising phase of the action potential.
And this is an all-or-none event.
Completely. Once you hit that threshold, it fires at its full stereotyped amplitude. There are no half measures. It either fires completely or not at all.
And then the signal has to travel down the axon. And this is where the genius of myelination comes back in.
This is where it all pays off.
Mm-hmm.
The speed of that propagation is all about saltatory conduction. In an unmyelinated axon, the action potential has to be regenerated at every single point along the membrane. It's slow and it's incredibly energy intensive.
But myelin changes the game.
Completely. The myelin provides this high-resistance insulation that prevents the current from leaking out. So the electrical wave is forced to jump from one node of Renvier to the next, only being regenerated at those specific points where the sodium channels are clustered. This makes the signal travel exponentially faster and more efficiently.
Now, you mentioned that the switch from CNS myelin to PNS myelin is the key anatomical point. Let's touch on 6.2, the CNS-PNS transition zone.
Yes, this is a really interesting and vulnerable spot. It's the precise physical boundary where the oligodendrocytes of the CNS stop and the Schwann cells of the PNS begin.
Where does this happen?
It typically happens right where the nerve roots are exiting or entering the spinal cord or brainstem. And it's not just the myelinating cell that changes. It's also where the brain's own protective layer, the glia limitans, gives way to the PNS's protective perineurium.
So it's a major handoff point. Why is it vulnerable?
Because the immune system recognizes CNS myelin proteins differently from PNS myelin proteins, and the nature of the protective barriers changes, This zone can be a site where the nerve is more exposed to immune or inflammatory attack, making it susceptible to certain disease processes. It's a chink in the armor, so to speak.
Okay, finally, let's compare the two final output points.
Yeah.
6.3, the neuromuscular junction versus the autonomic motor terminations.
A fantastic comparison because it highlights specialization. Let's start with the neuromuscular junction, or NMJ. This is the somatic output that controls your skeletal muscles, and it is specialized for two things: speed and absolute reliability.
What does it look like?
The axon terminal forms this specialized structure called the end plate, which sits in a little trough on the muscle fiber, and the muscle membrane underneath it, the postsynaptic membrane, is thrown into these deep junctional folds.
Yeah, those folds are packed with receptors.
Densely packed with nicotinic acetylcholine receptors, and the key functional feature here is what's called a high safety factor.
What does that mean?
It means that a single action potential arriving at the terminal releases a huge amount of acetylcholine, far more than is actually needed to trigger a contraction. This ensures that every single signal from the motor neuron reliably, without fail, causes the muscle to contract. There's no ambiguity.
It's built for non-negotiable activation.
Exactly. Now, contrast that with the autonomic terminations that are controlling your smooth muscle or your glands. They are much less specialized.
How so?
The axons are often unmyelinated, and they don't have a single defined end plate. Instead, the axon has these swellings along its length called varicosities.
And the neurotransmitter is released from there.
Yes, it's released from these varicosities diffusely over a wide area of the target tissue. The transmission is slower, it's less precise, and it's much more modulatory in nature.
And the distance to the receptor can vary.
Dramatically. The neurotransmitter might have to travel anywhere from 5 nanometers to, in some cases, 20 millimeters, diffusing through the tissue to find its target. This stark contrast, the precise high-fidelity NMJ versus the diffuse modulatory autonomic synapse, perfectly reflects the difference between needing to make a rapid, precise muscle movement and needing to gently regulate the secretion of a gland over time.
To wrap this all up, let's bring it back to pathology with 6.4, some clinical insights. When any of these incredibly specialized components we've discussed breaks down, the consequences are clear.
They are, and the most profound example is demyelination. The loss of that myelin sheath, whether it's in the CNS and MS or in the PNS, has a devastating effect on conduction.
What happens to the signal?
The depolarization current, which is supposed to be funneled to the next node, now spreads out and leaks through the bare axon membrane. Because the sodium channels needed to regenerate the signal are only clustered at the nodes, that weakened current can't reach the next node with enough strength to hit the threshold.
So the signal just dies out.
It just shorts out. It leads to conduction block, or at the very least, extreme slowing of the signal. And that is why patients with demyelinating diseases experience such profound loss of motor and sensory function. The wires have lost their insulation.
And the sources also give a very clear example with herpes zoster, or shingles.
A perfect example of a pathogen exploiting the neuron's own biology. The chickenpox virus travels up the sensory nerves and then remains latent, dormant for decades inside the sensory ganglia, like the dorsal root ganglia.
It's using that retrograde transport highway we talked about.
Exactly. And then, often triggered by stress or a weakened immune system, the virus reactivates. When it reactivates, it starts replicating and causes massive damage to both the neurons and the surrounding glial cells in that one ganglion.
And that leads directly to the symptoms.
Directly. The inflammation and death of the neurons in that specific ganglion is what causes the severe pain and the characteristic rash that appears in the dermatome, the strip of skin supplied by that single, afflicted sensory nerve root.
So, what does this all mean? We've gone from the grand map of the nervous system all the way down to the precise location of a potassium channel and the proteins that seal a 20 nanometer gap.
And I think the big takeaway is that the system's incredible speed and precision aren't just some monolithic electrical property. They are the direct result of highly specialized anatomy, the myelin, the nodes of Ranvier, and that anatomy is supported by this incredibly localized, tightly controlled chemistry at the synapse.
And none of it works without the support crew.
Not for a second. The system is entirely reliant on the glia. They're not just feeding and protecting the neurons, they are actively participating in the conversation. The concept of the tripartite synapse proves that. Without astrocytes cleaning up glutamate, without the molecular seals at the paranodes, the whole system just collapses into a toxic Schultz-circuiting mess.
We've really seen how dynamic this whole architecture is. The dendritic spines are constantly being remodeled by experience, which is the physical basis of learning. The Golgi tendon organ is mediating this life-saving reflex. It's a system in constant flux.
It is, and that leads to a really profound final thought. We know that even tiny changes in the shape and placement of these microscopic structures are what underpin memory, but also what goes wrong in aging and disease. We see the loss of spines in Alzheimer's, the failure of myelin in
So
MS.
the question is,
The question is, if the neuron is the computer chip, then the glia are the engineers, the power supply, the cooling system, and the maintenance crew all in one.
yeah,
How much of our conscious identity, our ability to learn, our resilience against disease is fundamentally determined not just by the neuron firing, but by the health and dynamic activity of these sophisticated support structures that we once dismissed as mere glue? That relationship between the star and the stage crew is still one of the deepest mysteries we're trying to unravel.